Hello friends, and welcome to our video lecture on topic B3.3, muscles and motility. This topic is additional higher level content, so if you are an SL friend, you will not be assessed against this material. Our guiding questions: how do muscles contract and cause movement, and what are the benefits to animals of having muscle tissue? Our objectives for today: we're going to talk about some adaptations and our needs for movement. We're going to explain the sliding filament theory of muscle contraction. We're going to talk about the structure and function of titin and identify the structures of a motor unit. We're going to describe the interactions between muscles and skeletons and talk about antagonistic muscle pairs. We're going to identify the structures and functions of a synovial joint, talk about how we can measure range of motion in our joints, and then describe adaptations for swimming. Our focus is definitely going to be on muscles as an adaptation for movement, but know that there are definitely lots of organisms that move that don't have muscles. And in fact, we have organisms that are considered sessile. They're kind of stuck in place, but they can still move. So this Venus flytrap is sessile. He is stuck here. His roots are embedded in the ground. It cannot run or crawl away, but it does still move. When a bug lands on its leaves, those leaves will close in response. That movement allows the plant to capture that animal, that insect, so that it can digest it for the nitrogen. We're going to talk more about motile animals like this lovely sloth. He doesn't move fast, but he definitely does move. Why do organisms need to move? Our needs for locomotion include foraging for food. Of course, we need to go and find snacks. Escaping danger. If you are prey, you want to get away from that predator. We definitely move to search for mates. Migration, whether it might be seasonal or maybe related to looking for food, migration is a very important need for movement. And then dispersing. We don't necessarily all want all of our babies to stay right under us all the time, and so spreading out our species is an important need for locomotion. Are you ready for so much vocabularies? So here we've got a bone. This bone is attached to a muscle using a tendon. Tendons are connective tissue that connect bone to muscle. That muscle is composed of lots and lots of muscle cells. Muscle cells are weird. They're very large. They're multinucleate, but they are still cells. They've got cytoplasm. We actually call it sarcoplasm, but it's still cytoplasm. They've got lots of mitochondria, but they also have lots of nuclei, so that's kind of weird. We actually call our muscle cells muscle fibers. So muscle fiber is the same thing as a muscle cell, even though it's an atypical cell. Inside our muscle fibers, we have that cytoplasm and some mitochondria and lots of nuclei, but also we have these long strands, these chains of proteins called myofibrils. And we are zooming in on one myofibril here. So these myofibrils are composed of lots and lots of bundles of actin and myosin proteins. Actin and myosin proteins. We're going to zoom in on one of these tiny little pieces of this bundle, this myofibril that's in a muscle cell, muscle fiber, that's in a muscle that's attached to a bone with a tendon. We're going to focus in on this super quick. So a bundle of actin and myosin. This bundle of actin and myosin, here's some actin and here's some myosin. Our actin, our myosin. Actin filaments, myosin have these super cool little divots. We're going to call them myosin heads. In this functional unit of a muscle, which is called a sarcomere, sarcomere, this functional unit of a sarcomere, we have kind of ends, and we call those ends the Z discs. Z discs are the ends of the sarcomere. So Z disc here, Z disc here. The Z disc is attached to the actin. Again, we've got our actin filaments in blue. We have our myosin is here, myosin in red. The myosin is attached to the Z disc with another protein that we're going to talk about in a couple slides called titin. And what's pretty cool is that these myosin heads can bind to the actin filaments and pull them. And so what happens is you can see this H zone here, When those myosin heads bind to the actin filaments and pull them, that H zone gets smaller because the myosin pulled those actin filaments closer to what we call the M line in the middle. And notice that my Z discs are now closer to each other. The whole entire sarcomere is shortened during contraction. It's a lot to think about, right? Let's look at this in a little bit more detail. And you're probably thinking, more detail? Yes, my friends, more detail. So again, here we've got our bone. We've got the tendon that attaches the muscle to the bone. Muscle has muscle cells called muscle fibers. Inside those muscle fibers are these myofibrils, these chains of protein strands, myosin and actin, and that little bit of titin. The functional unit of these chains of myofibrils is called a sarcomere. Sarcomeres go from Z disc to Z disc. Again, we've got our actin, we've got our myosin, the myosin heads here, myosin attached to the Z disc with that titin. We're going to focus in on one tiny little piece here, this actin, this myosin head. And we're actually going to start at number 3, because I like to start at number 3, not at number 1 in this diagram. So we have our myosin head, we've got our actin, we've got our myosin head, myosin head, and here's our actin. ATP is going to bind to the myosin head, and that allows that myosin head to get ready to bind to the actin filament. The ATP is going to hydrolyze. Remember that hydrolysis is splitting things using some water. That ATP is going to pop apart into ADP and inorganic phosphate. So that hydrolysis of ADP, inorganic phosphate, that allows for what we call the cocking of the myosin head. So it's ready to bind. The inorganic phosphate is actually going to pop off. This diagram doesn't do a very good job of that. So that inorganic phosphate group actually pops off, and that allows our myosin head to stick to the binding site on the actin. We call this a cross bridge. And then the ADP also pops off, and that causes the myosin head to bend. We call that a power stroke, and that pulls that actin toward the middle where the M line is, shortening the H zone, shortening the whole entire sarcomere. Feeling good? The next slide has the same stuff in just words, so hold on just 1 second if you're like, better? Okay, so here again in words, our myosin heads move into the cocked position when ATP turns into ADP and inorganic phosphate. That gets that myosin head ready to stick to the actin filament's binding site. When that happens, that's called a cross bridge, and that inorganic phosphate is released. And then the ADP is also released, and that allows those myosin heads to bend. We call that the power stroke, pulling the actin filaments to the center of that sarcomere toward the M line, shortening the H zone, shortening the whole entire sarcomere, which contracts the muscle. And then another ATP molecule comes in, binds to that myosin head. That myosin head is then released from the actin so that it can do it all over again. Here we have some lovely images of relaxed and contracted sarcomeres. Again, we've got our Z disc, so the whole thing is a sarcomere. Myosin, actin, myosin heads. This is relaxed. Notice how nice and wide is my H zone. That H zone, so nice and wide. This is a micrograph of actual muscle tissue. We've got our Z disc. This is going to be all of our myosin. This is where I've got just that actin. So this part is here. So just actin. We call that the light zone because it's lighter in color because that actin protein fiber is a little bit thinner than the myosin. We're going to call this our dark band. It's the dark band because it's lots of myosin, which is that thicker protein filament. When it contracts, again, we've got the ATP. It hydrolyzes, causes those myosin heads to get into the cocked position. Inorganic phosphate pops off so that it can bind. The ADP pops off, and the whole thing goes, and it squeezes those actin filaments together. Notice that my H zone here is very small. My H zone is quite a bit smaller. Notice my Z discs here and here are closer to each other. The whole entire sarcomere is smaller. Again here, Z disc, There's that M line. The H zone is so tiny now. My dark band is a little bit smaller just because that H zone is skinnier in the middle. Notice that the light bands are also quite a bit thinner. Light band here compared to the light band here. So relaxed, contracted, feeling good. Let's talk about titin now. So titin, again, is that protein that binds the myosin. Here's my myosin to the Z disc. Here's titin. Titin is super cool. It's like this little spring protein. It is a giant protein. It is composed of 34,000 plus amino acids. It is a protein with quaternary structure. Remember that quaternary structure means that there are multiple polypeptides held together, perhaps with some disulfide bridges, some hydrophilic interactions. So this quaternary structure, like hemoglobin, it's got those 4 subunits. Titin has 244 subunits. Oh my goodness, it's ginormous. It is an enormous protein. And it acts like a spring in our muscles. So here it is, kind of zoomed in. Here's our titin. Here's our myosin. And that myosin, again, is bound to the Z disc with titin. So titin is this orangey yellowish guy. Z disc myosin. And you can see here that it's kind of contracted. And then here it gets pulled out. So as the Z disc is pulled in toward the M line as those sarcomeres shorten during contraction, this happens. So we've got our super short pieces of titin. And then when the muscle relaxes, the myosin doesn't change in length. The actin doesn't change in length, but this titin can stretch back out. So muscle relaxed and then muscle contracted. Let me erase so that you don't have all those squiggles. Again, muscle relaxed. Notice how that titin is all stretched out so that the actin and the myosin are not overlapping so much. But then here we have that titin is super compacted so that our actin and myosin can have all that overlapping. And this is why we call it the sliding filament theory. These filaments of protein slide over each other. And when the muscle is contracted, there's lots of overlap. And then when it's relaxed, there's not so much overlap. The sliding of those filaments, the contractions of our muscles, depends for the most part on some nerve stimulation. Now, if we're talking about the heart and we're myogenic, we've got our sinoatrial node. But if we're talking about like the muscle in my arm that I'm using right now to draw on the drawing tablet here in this video lecture, then I have motor units. Motor units are nerve bundles that talk to the spinal cord, and the spinal cord talks to the brain. Signals are sent along these motor neurons. It's called a motor neuron because its job is to cause the muscle to move. The axon is this crazy long tail-like structure. We'll talk about that in a little bit when we get to our lesson on nerve cells. These guys can have lots of branches, and then they end in what we call these neuromuscular junctions. So this is the end of the nerve cell, and then here is some muscle cell, a muscle fiber. Neurotransmitters are released from the end of that motor neuron. Those neurotransmitters will bind to the muscle fiber, and then all the magic happens. So motor units, how we control our skeletal muscles. We've got our motor neurons. We've got our neuromuscular junctions that talk to, and generally they talk to many of those muscle fibers at the same time so that we have lots of our muscle tissue contracting together to make our big movements. We've been talking about these amazing muscles, but muscles are important because most of the time they're moving bones, and that allows us to have that movement. We have different kinds of skeletal systems. We, of course, have bones on the inside. That would be called an endoskeleton. But there are lots of animals that have exoskeletons, external skeletons instead. Our exoskeleton animal friends, what I love about this, so the exoskeleton is composed mostly of a compound called chitin. Chitin is also what makes up fungal cell walls. So fungus and exoskeleton have a lot in common. It's pretty cool. But what is similar between endoskeletons and exoskeletons is that muscles move them. For us, our endoskeleton muscle is going to bind to the outside of our bones and contract and move those bones. What's pretty cool about the exoskeleton animals is that their muscles actually connect to the insides of their exoskeletons and then can bend things around like that. Our bones act a little bit like levers. So we've got a fulcrum, which would be the joint. And then when the muscle contracts, it pulls that lever up. And lots of physics words about the fulcrum and the effort and the load. And we'll talk about this a little bit in class when we do a lab with our joints and our muscles. How our muscles move those skeletons around for the most part is through the use of antagonistic muscle pairs. We know from literature class that antagonists are like against the protagonist, they're the bad guy or the thing that's causing conflict. Here, it's not so much conflict as it is simply opposite directions. So here we've got our arm. My arm is extended because my triceps are contracting. The triceps are our extensor muscles. When my biceps instead contract, what happens is that shortening of the muscle pulls the arm up. And so now I have a flexor or flexion of the arm because that biceps is contracted. But when the biceps is contracted, my triceps has to be relaxed. But then if I want to straighten my arm back out, my biceps can't push the arm down. Instead, I need to contract the triceps, and the triceps, when this shortens, it pulls that arm back down while the biceps are relaxed. So antagonistic because they're working in opposite directions, but if they weren't, we wouldn't move very well at all. What's kind of cool about these antagonistic muscle pairs, clearly I'm looking at an endoskeleton here, but our exoskeleton insect friends have the same. They have antagonistic muscle pairs on their, well, inside their exoskeletons. We have actually already talked about an antagonistic muscle pair, my friends. Our external and internal intercostal muscles are antagonistic muscle pairs. We have our ribs. The ribs are the costal bones. We have muscles between them. That's the interpart of the costal, intercostal muscles. We have some that are external and some that are internal, some that are on the outside between the ribs. Here, closer to where our skin is, we have some that are internal between the ribs, closer to where the lungs are. And what happens when the external intercostal muscles contract, our ribs get pulled up and away, and we inspire or inhale. And when our internal intercostal muscles contract, what happens is those ribs get pulled closer to each other and closer toward the thoracic cavity, and we increase the pressure, decrease the volume, and air goes out. We expire. Good. We've been talking a lot about how these muscles move, how muscles pull on bones so that our bones can move, but where bones meet other bones is known as a joint. And some of these joints are synovial joints so that our bones do not grind against each other because that would not feel good. So here we're going to focus in on a specific joint, the hip joint. Here's pelvis, here's femur, one of the bones of our legs, the biggest bone of our leg, and we're focused in this picture on this spot of our hip joint. Again, we've got the femur. The femur has a super cool shape that has this like big ball-like structure, so we actually call this a ball and socket joint, where that top of the femur makes the ball and the pelvis makes the socket. So that we don't have bone grating on bone. Some of the adaptations we have in here of our synovial joint include a synovial membrane, which encloses our joint capsule. So the synovial membrane is going to keep that capsule closed. Inside of that synovial joint capsule, we have some synovial fluid. And that fluid acts like a layer of oil that just helps to keep things lubricated so that the bones can slide against each other instead of grind against each other. We also have some cartilage in here. That cartilage also acts as a cushion between the bones. It's kind of squishy, so it helps with impact. So again, these synovial joints, this one happens to be the hip joint where it's a ball and socket. we've got our femur, we've got our pelvis. We have this synovial capsule enclosed by synovial membrane. Inside it is that synovial fluid that helps to lubricate so that this joint can slide instead of grind. We also have cartilage, which is kind of squishy to help with that impact. We are also looking here at another kind of connective tissue. So we talked earlier about our tendons, the tendons that bind our bones to our muscles. But there's also connective tissue that binds bone to bone, and that's called a ligament. So ligaments are bone to bone, and tendons are bones to muscle. How I remember, L comes before T in the alphabet, and B comes before M in the alphabet. So bone to bone is a ligament, bone to muscle is a tendon. We can actually measure the range of motion of our joints with this super cool device known as a goniometer. It is basically a protractor for body parts. So we take one arm of the goniometer and we line it up. In this case, we're looking at a femur. We put the rotating part, the protractor part, here on the knee. And then as the patient bends the knee, we can bend this side of the goniometer with it. And so we can measure from flexing to extending the range of motion with this protractor-like device. So we humans are pretty cool. And talking about synovial joints and goniometers is really cool because it's about us. But there are other animals that are also super cool that are swimmers. And so here we're going to look super quick at some adaptations for the movement of swimming. Our swimming friends have streamlined bodies. They are, I guess, not aerodynamic, hydrodynamic, just to help them propel through the water a little bit more smoothly. They have very little hair to reduce the drag in the water. Their front legs have evolved into flippers. So instead of having arms and hands, they have these lovely flippers. But do notice that they still have finger bones inside their flippers. That's so cool. They don't have any hind legs. Those hind legs have evolved away. They do have tiny little pieces of pelvis bone left, though, just floating around there. They've got a fluke. This fluke is this horizontal fin at the end, and its job is to flap up and down again to help with that propulsion. They also often will have a blowhole, which closes up super tight, and then they can hold their breath really, really, really for a very long time. And on that note, my friends, we have talked about muscles and motility. We outlined those adaptations for movement. We talked about the needs for movement. We might need to find a mate. We might need to find some food. We might need to migrate. We talked about the sliding filament theory of muscle contraction. We've got our myosin heads that bind to the actin, and then there's the power stroke when the ADP pops off, and that causes the actin to slide toward the M line, shortening the sarcomere. We talked about the structure and function of titin. It's ginormous, and it acts like a spring. It binds the myosin to the Z discs. Those motor units, those, remember, are our motor neuron, that nerve cell that talks to muscles, that neuromuscular junction where neurotransmitters are released from the neuron, bind to the muscle fibers to get those muscle fibers to contract. We talked about interactions between muscles and skeletons. Some of us, like us humans, have endoskeletons. Other things have exoskeletons. The muscles stick to the skeletons and move them. Muscle is bound to bone with tendons, bone to bone with ligaments. We talked about antagonistic muscle pairs like our internal and external intercostal muscles. We talked about synovial joints. There's the membrane, there's the fluid, there's the cartilage to help to prevent bones from grinding against each other. We have some goniometers. We're going to play with them in class. And we also talked about those adaptations for swimming. Great work today.